So the organism's abundance as well as their composition is going to give us a clue as to what the climatic conditions were at their time of burial. Some organisms can only live during warm times, for example. So if they are abundant at a certain thickness of sedimentary rock, then the climatic conditions were warm and favorable for their growth and reproduction during the whole sedimentation of that stack of rocks, that thickness of it. And vice versa, right? So for organisms that liked colder climates, for example, if they are thickly deposited, then that means that the conditions were favorable in that respect. The same thing goes for their lack of widespread conditions. So if there was a short amount during which you could find them, a short layer, then that means that probably the conditions were changing relatively fast and they couldn't adapt to a colder or warmer climate depending on the organism. The amount of sediment that was burying the organisms or that was near their abundance span also gives us a clue about the environmental conditions that were needed for the sedimentation rate that we found on each of those sediment cores. So if, for example, we could find a relatively high amount of organisms in a sediment core that was full of sediment, so let's say that there was a whole meter of sediment of mudstone, then it is possible that the sedimentation rate of that mudstone was high. But if we find the same number of organisms over just, say, a meter of mudstone, then perhaps the sedimentation rate was a little bit lower, even though the organism could live in the same span of time. So, in much the same way that we use the stable isotope analysis for the delta 016 to 018 in the ice cores, we can use it for understanding the temperatures during the time of formation of the marine organisms. So remember from chapter 18 and 15 that isotopes are different representations of atoms from the same element. So these will have a different atomic mass due to the number of neutrons that they have in their nucleus. So again, oxygen in its more common form is going to have 16 as a mass of 16. However, there are two other stable isotopes, which are going to be oxygen 17 or oxygen 18. In which case oxygen 17 is going to have one more neutron in its nucleus, and oxygen 18 two more neutrons in its nucleus. Out of the two, oxygen 18 is the more common. So overall, oxygen 16 makes up 99.78% of the atmosphere, or the oxygen within the atmosphere and within the crust, water, all of that, all of the spheres. About 0.2% is going to be made up from oxygen 18, and then the remaining goes for oxygen 17 pretty much. So because the atomic mass of oxygen 18 is so much greater than that of oxygen 16, it is going to be overall heavier than oxygen 16. And so it's going to be harder to lift because of its density, because of how heavy it is. So when evaporation of a liquid or of water itself happens, the H2O that makes up water is going to become gaseous and is going to be lifted up onto the atmosphere. And as we know, evaporation is enhanced during times of warmth when we have high temperatures. So high temperatures tend to lift up the heavier isotopes as well. Again, so when temperatures are higher, there's more heat, and hence there's going to be more energy that's applied to the system, which will then evaporate more water. With this greater energy, there's also a greater potential to lift, again, those heavier oxygen 18 atoms, as well as the oxygen 16 ones. But overall, that means that the quantity of oxygen 18 inside the ocean is going to be reduced on average as it is being lifted up with the warmer climates. Okay, so an organism's hard parts, which again are things such as shells or exoskeletons, tend to be made up of calcium carbonate or of silica at times. Silica is SiO2. These organisms use the available atoms in the ocean waters to make these hard parts. So, if the stable isotope analysis of oxygen 16 to oxygen 18 is done for the fossil itself that is made out of calcium carbonate or silica, and the ratio of this oxygen 16 to oxygen 18 is higher than the average today, then the temperatures were warmer than today's, because much of the oxygen 18 that was in the ocean was evaporated by the high heat potential. If, on the other hand, the ratio of oxygen 16 to oxygen 18 is lower than the average today, then the temperatures were colder than today's because there was not enough heat to lift those heavier oxygen 18 atoms from the ocean waters, and so they remain and make the ratio lower. So, overall, the composition of these organisms, the organisms' hard parts, is dependent on the temperature of the environment during the time that they lived. Another form of proxy data is glacial ice. So we discussed this quite a bit when we covered chapter 15, but we'll talk about it briefly still. So scientists collect the samples of ice or these ice cores by using a drilling rig, which is pretty similar to an oil drill, but it is much smaller. Some of these samples that they have collected of ice cores measure up to 3,000 meters from the Antarctica ice sheet and allow us to have about 700,000 years of climatic history, which is going to be a proxy data again. So once these ice cores are recovered, we can count the small layers that make up the ice, and it can tell us basically the number of years that the ice has recorded history for. Also, we can do stable isotope analysis in much the same way that I described in the last slide. And in this case, it is the oxygen used by the molecules of ice rather than the oxygen used to make up the hard parts of the organisms. So, in this case, if we use the stable isotope analysis, a larger oxygen 16 to oxygen 18 ratio than today's average is going to signify that the temperatures were colder than today's, and a smaller oxygen 16 to oxygen 18 ratio than today's average is going to signify that the temperatures were warmer than today's. Another analysis that can be done on the ice cores is checking the atmospheric composition at the time of each layer's formation. So the tiny pores which lie in between the ice particles were basically captured during the formation of the ice layer, and they will contain a signature of the atmospheric conditions during this time of formation. So different greenhouse gases concentrations can be measured within those pores to check for past temperatures. So as I mentioned in chapter 15, many greenhouse gases concentrations are actually positively correlated with temperature. So the greater the concentration of greenhouse gases that we find in these tiny pore spaces between the ice, then the warmer that the temperature was at the time during which the ice layer formed. All right. Trees, as some of you may know, when they grow, their trunks get larger. They get thicker. And it gets larger due to the addition of material to the trunk. So the addition of material occurs through time, and it's typically seasonally controlled. So every time a year goes by, a new ring is added to the tree's trunk. Each ring is going to represent a period of rapid growth, which in the image appears as the light-colored parts. And this rapid growth typically takes place during the spring and the summer months. And you're going to have a period of slow to no growth, which in the picture is represented by the dark part of the ring, and it typically happens during fall or winter times. However, as you can see in the image, not all of those rings have the same thickness, the same length. Some of them are much larger than others. The thicker rings represent more favorable conditions for growth, which, depending on the tree species, it may mean that the temperatures were warm or they were cool, or the conditions were wet or they were dry. Or it could be a combination of these factors, depending on the species of trees in which you're measuring the tree rings. And then again, thinner rings will represent unfavorable growth conditions. So by counting the number of rings, as well as measuring their thickness, we can have an idea of the climatic and environmental conditions during the lifetime of that tree. And if the trees that are around the area also show a similar pattern, then there is a further certainty that these ideas that we initially got are correct from the one tree sample. So this study of tree ages and rings is called dendrochronology, by the way, and in the University of Idaho has a really cool dendrochronology lab. If you're ever interested, please let me know. I'd be happy to refer you to it. I believe that this lab is connected to the geography department, but dendrochronology is useful for many other disciplines, such as geology, as I just told you, climatology, ecology, or archaeology, for example. All of these have a use for dendrochronology. A few other sources that provide us with, again, clues to decipher these paleoclimatic conditions also exist, like fossil pollen study, which is called palynology. So palynology literally means study of pollen. So climate is a major factor that influences the overall distribution of vegetation, and the nature of the plant community occupying an area is also a reflection of the climate. So pollen and spores, which are part of the plant's life cycles, due to their resistant walls, tend to be preserved in the sediments much better than some of the other parts of plants, for example. So if we analyze the pollen abundance and we decipher the species of plants that the pollen is from, then the climatic conditions for the time that said pollen was preserved can be inferred. Another proxy data are coral rings. So corals, which are invertebrate animals that live in warm and shallow waters in the sea, build their skeletons from calcium carbonate or calcite, which is extracted basically from the ions that are dissolved in the seawater. Because the skeleton is made out of calcite, stable isotope analysis can be done on the coral rings themselves. So, like trees, corals will also exhibit growth rings, which are based on seasonal variations as well. And these are distinguished based on the density differences between a time that was favorable for growth of those rings and a time that was not favorable for growth of those rings, also again controlled by seasons. Lastly, we also have historical documents, which sometimes will contain helpful information. So most historical documents don't tend to mention the relatively stable or constant atmospheric conditions, but they will mention events that are out of that common rule. These events can be things such as floods or volcanic eruptions, blizzards, maybe the migration of animal species, including themselves, and they can include things such as crops and their success or lack thereof. So overall, by these accounts in the historical documents, we can, or it can provide us with this useful evidence of some possible influences from the changing climatic conditions at the time that the document was made.